Hello and welcome to our YouTube channel. From today onwards, we are going to start our new series, which is the biology series. Many of you have requested us to create a new series on this biology thing based on the exam pattern and everything. So we'll be dealing with the basic biological sciences, which is important for our exam point of view. So I hope that, like our history lessons, we will be succeeding in delivering you great content for your exam purpose, and you'll be liking us and will be supporting us till the end of this series. So we are going to divide this whole series into several parts. We are starting with basic cell theory and everything related to cell, which is mainly essential. Not everything means we won't be doing the PhD regarding cells and everything over here. We will be only covering those parts of the cell theory which is important for our exam perspective only. This is the same thing which I am repeating in every series, that you have to read only those parts which are essential. You don't have to read everything under the sun in order to get a job, because that is not required here. If you want to study on your own, that's up to your choice, you can, but we will be covering only those portions which are essential to our syllabus. Anyway, we are going to start with the cell theory and cell division theory. So let us start, and I hope that you will be enjoying this video. And if you have any kind of suggestions, then definitely give us the feedback in our comment section, or you can mail us to our email address, which is given in the description link as well. Okay, so let us start our discussion regarding this. যদি আমাদের এই ভিডিওটি আপনাদের ভালো লেগে থাকে তাহলে অবশ্যই একে লাইক, শেয়ার এবং সাবস্ক্রাইব করবেন এবং আমাদের পাশে থাকবেন আমাদের উৎসাহ দেওয়ার জন্য। Okay, so let us first discuss the features of living organisms. How will we define a living organism? How can we differentiate it with an non-living organism? So there are some basic features which defines a living organism. Those are, first, it has a definite shape and size. Even the non-living organisms do have, but they don't have a definite shape at the very innermost level. Secondly, metabolism. What is metabolism? We all know what is metabolism. We have already read these things in our school days also, that the metabolism rate of a person and everything. What is that? That nourishment obtained from organic compounds. Like you are eating something and your body is extracting the needy things from that food item, and those food items are building your body. So this is metabolism. Anabolism, process to increase protoplasm. Then catabolism, process to release energy. Then growth and development. Reproduction by asexual or sexual means. Response to stimuli, and adaptation to survive. So these are some basic features of a living organisms. Like these are the things which define that yes, you are a living being, or a thing is a living thing. And based on those, various researches have been done, and the scientists, they have shown the way how to define a living thing and what are the basic unit of a living thing. Moving on to the basic unit of a life, that is cell. Now what is a cell? Cell is nothing. Cell is basic unit of a living thing. Like how do we define cell and everything? That you can divide cell into other molecules and everything, but cell is the last functioning unit of a living organism. If you divide cell, and if you just take out the inner structures of the cell, they cannot work on their own. Cell is the last functioning material of a living thing. That is why this is called the basic tenant, or the basic unit of a life. Though there were several other theories of cell and several other researches on cell, but the cell theory by Schleiden and Schwann, these two German biologists, that has been popularized, and we take those theories, those tenets of the theory as the essential of cell theory. What are those? All organisms are made of cells. All the living organisms which you are seeing over here in this planet are made of cells. Second, cell is the basic unit of structure and function of all organisms. Just what I told a few minutes ago, that this is the basic unit of structure and function as well. If you divide a cell, the living organism cannot function. Third tenet, all cells have their own biosynthetic machinery. All cells have their own machineries inside them so that they can work properly, and we will be just reading all those in upcoming minutes, in upcoming slides as well. We'll be showing you how the cell works and everything, so stay with us. And the last tenet is all cells arise from pre-existing cells. Okay? A cell cannot come out of nowhere. There should be, or there must be, a mother cell from where the cell comes. These are the basic tenets of the cell theory by Schleiden and Schwann. Now there are two types of cells according to the researchers and everything. There are prokaryotic cell and eukaryotic cell. Then what are those? Let us just very simply discuss this thing from the exam point of view. First, let us take the prokaryotic cell. What are these? Well, every cell has a nucleus. Okay, and nucleus is the main epicenter of a cell, you can say. So in prokaryotic cell, there is no membrane of a nucleus. Like a cell's body is made of so many things. Like if you consider a jug full of water as a cell, and if you just give some of the spices into the jug, and the biggest spice which you have put into the water, if you consider that as a nucleus, then that nucleus is separated by a membrane with the other parts of the cell. So in prokaryotic cell, what happens? That there is no definite membrane of a cell. You can see in the picture that the nucleus of prokaryotic cell is so disheveled. There is the absence of a particular membrane which can define the boundary of a nucleus over here. The second feature of prokaryotic cell is organized nucleus is absent. As there is no membrane of the nucleus, definitely the nucleus is not organized, it is disheveled. Third thing, it is naked. The DNA of the cell is naked. Like in eukaryotic cell, you will find that DNA is covered with all those kind of ER and everything, which we'll be discussing later on. But in this case, the DNA just moves on into the cytoplasm. Cytoplasm means the kind of liquid in which all the other elements of the cell remains. So the ribosome just floats over there, and the DNA also floats over there. And lastly, no membranous organelles. As I told you, that there is no such distinctive elements or organelles of a cell. Now the question may arise, what is an organelle? Organelle is nothing but the parts of the cells or the elements of the cells which consists of a cell. Like a mitochondria is an organelle, the the chromosome is an organelle, then the ribosome is an organelle, the DNA is an organelle, then the nucleus is organelle. So these are all the things which are organelles. Like, as I have given you the example of a jug full of water, all the spices which you have given in the jug are the organelles of the cell. If you consider the jug as a cell and the water as a cytoplasm, then all the other elements which you have put inside the jug are organelles. Now let us move on to the eukaryotic cell. Well, eukaryotic cell, I'm sorry, I didn't give you the example of prokaryotic cells. Those are mainly bacteria and blue-green algae. Basically, those one cell living organisms which are the basic elements of life, which had been and still are the basic elements of life, those are the prokaryotic cells. Means their cells are not that much of developed. Now we have the eukaryotic cells. What are those? What are the features? Whatever the prokaryotic cells have, we have the opposite of that. Like, we have a well-defined nucleus. The nucleus is very much defined and very in a good shape and size. Then we have a cell wall. The eukaryotic cell, they have a cell wall. Though the cell wall is absent in animal, we have the cell membrane, but in plant you can find the cell wall and the plasma membrane to enclose the protoplasm. So there is a membrane in the cell which actually gives a shape and a size, and also helps the cell to carry on with more complex functions. And lastly, cytoplasm with both living and non-living inclusions. It means in the cytoplasm, which is the fluid inside the cell, is both absent in living inclusions and non-living inclusions as well. Now let us move on to the topic of the difference between plant cell and animal cell. These are the very basic differences which can be asked in the question. So just pay attention to this portion. Even UPSC in 2020 also have asked questions from this portion. So what is a plant cell? A plant cell has a rigid cell wall, whereas in animal cell the cell wall is absent. We have cell membrane. Plant cell cannot change its shape. Animal cell can change its shape often. Plastids are found. Plastids are usually absent. There are plastids in very few animal cells which can do the photosynthesis on their own. These are you can say those are exceptional. Plant cell contains a large central vacuole. Animal cell contains many small vacuoles. Plant cell has comparatively fewer mitochondria. In animal cell, mitochondria are more numerous. In plant cell, lysosomes are rare. In animal cell, lysosomes are found. So these are the basic difference between two kinds of cells. And from here, I again repeat, the questions have been asked. Anyway, so you can see the structure over here in this slide. We have given a picture over here, then how the plant cell and the animal cell looks like. Though these are all 2D pictures, but actually you have to imagine in 3D. In that case, you can go to the NCERT book and you can read the chapter from there. They have a very good illustrated and 3D pictures over there in the book, and you can just compare your study or your knowledge with those pictures in order to get a better understanding of the subject. Anyway, now we'll be moving on to the organelles of a cell, like how a cell works and what are the structures, what are the elements of a cell which contains in it, and what are those. So let us start. The first thing is cell wall. Cell wall, we have already told you that this is not present in the animal cell, whereas in plant cell it is very much present. So this is a semi-rigid, freely permeable and non-living thing because it's a wall. It's a semi-rigid. These characteristics are important from your exam point of view. It's a semi-rigid, freely permeable and non-living, made up of cellulose, protects the cell and gives it rigidity. Definitely, that is why the name is wall, and supports the plasma membrane and the cytoplasm. Whereas what is cell membrane? Cell membrane is both present in animal cells and plant cells as well. So the cell wall is present only in plant cells, but cell membrane is present in both plant cell and animal cell. What is this? This is elastic, living and differentially permeable. What we saw in case of the wall, that this is semi-rigid and this is freely permeable. This is not the case with the membrane. This is elastic, this is living. The main difference, this is living and differentially permeable. Consists of 2 layers, whereas in cell wall we didn't see any kind of layers. Gives form and support to protoplasm. Definitely, that is why the membrane is there. Allows selective uptake of water and nutrients and removal of waste product. Like when you are drinking water or when you are drinking any kind of fluid which is essential to the cell, the membrane actually helps to just let pass the essential items or the essential things inside the cell and keep the wastes at bay. Okay, the next thing is cytoplasm. Cytoplasm is the fluid. If you take the analogy of the jug full of water, then the water is cytoplasm over here. What is cytoplasm? Part of the cell between the plasma membrane and the nuclear membrane. Okay, now if you go to the picture which we have given you after the slide of cell theory, you can see that there are 2 membranes. You just look at this the picture of the eukaryotic cell, that one membrane is in the nucleus, the nuclear membrane, and another is the outermost membrane, that is the plasma membrane. And in between those 2 membranes, the fluid which is existent in every way, in every other place, that is cytoplasm. Next, inner granular layer is called endoplasm, outer layer is called ectoplasm. Now in cytoplasm also there are 2 types. The inner is called the endoplasm and the outer layer is called the ectoplasm. Now the inner layer is granular, outer layer is somewhat clearer. And what does it do? Actually it contains organelles which are living parts of the cell, such as lysosome, mitochondria, etc. So these are the spices of the water. Lysosome, mitochondria, then RNA, DNA, everything, which are the living organelles of the cell. Cytoplasm holds all of them. It also contains a transparent homogeneous semi-fluid substance known as matrix. It also contains the matrix as well. This can be asked as a question, that which all of the materials or the living things does a cytoplasm hold? A, mitochondria, B, lysosome, C, matrix, D, nucleus. Then they'll be giving you a question, 1A only, 2B only, C, 1, 2 and 4 only, and D, all of them. The answer will be definitely all of them. Now the next organelle, which is endoplasmic reticulum. Now this is not a organelle like a thing, this is a kind of a network. Okay, what is this? This is a membranous network connecting nuclear membrane and the cell membrane. So this is a network through which the nucleus functions with the other cells, because this is a network which actually goes from the nucleus membrane to the outer membrane, forms the skeletal framework of the cell. We can say, like, these are all in a very hypothetical manner. You won't be finding any kind of skeleton inside a cell, but they forms a kind of skeletal framework, like how a skeleton works. Like it's a network of connecting your arm with your finger, your neck with your leg. So this endoplasmic reticulum does the same thing, works as a pathway for transfer of nuclear material from one cell to another, which I was telling you, that this actually connects another cell. This actually communicates with another cell, and that is how we grow our body. Rough ER is concerned with transfer of protein which are synthesized by ribosomes on their surface. There are ER like this endoplasmic reticulum which are rough and somewhat lucid also. This rough ER is concerned with the transfer of proteins. So from one cell to another cell, after a very complex synthesis of proteins by ribosomes, when you have to transfer the protein to another cell, you need this network. You need this endoplasmic reticulum network. Okay, the next organelle is ribosomes. Now, we have all known about ribosomes since our school days. Maybe we have scored well or we didn't score well during that time. Anyway, we have to read again it because we need to get a job. So yeah, what is ribosome? Ribosome is a dense spherical and granular particles that occur either on rough ER or endoplasmic reticulum or freely in the matrix. Now, matrix, I told you that in cytoplasm there are certain spaces where the fluid goes on according to its own wish, that is matrix. And ribosomes is also like the same. What is that? It is a dense spherical and granular particle which can consist either on the rough ER or it may freely traverse in the matrix. It is constituted by RNA, or the ribonucleic acid, and proteins. It is not bound by any other membrane, any kind of membrane, and present in both eukaryotic and prokaryotic cells. Except, these exceptions are essential for our exam point of view, except mammalian RBC, অর্থাৎ red blood corpuscle. Except mammalian RBC, this is present in prokaryotic and eukaryotic cells as well. And what does it do? Actually, it does synthesize the proteins. Okay. The next item is Golgi apparatus. Now, what does Golgi apparatus mean? Golgi apparatus, the name came from the scientist called Camillo Golgi. He actually first discovered this thing. So they are actually what? They are very flat disc-shaped sacs or cisternae of 5.5 micron to 1 micron diameter. Okay, so these are stacked parallel to each other. Okay, they stacked parallel to each other, and there is a they form a shape of a sac, and they stay near the nucleus. And what is the purpose of Golgi apparatus? They actually performs the function of packaging materials to be delivered either to the intracellular targets or secreted outside the cell. Arthat, they kind of do the packaging thing, that which thing should be inside the cell and which things shouldn't be inside the cell. You can say that they do the job of a chooser. They can choose. Okay. So where does it arise? It arises from smooth ear. Like ribosomes, they stay on rough ear, you can say. The Golgi apparatus, they arises from smooth ear. These are absent in bacteria, blue-green algae, Mechiosperms, and mammalian RBCs as well. Okay. Now, let us move on to the next organelle, which is important, that is lysosome. What are those? Sac-like structure. Okay, this is a sac-like structure surrounded by a single membrane. This is also a sac-like structure, that are lysosomes, and they have a membrane. This contains enzymes created by rough ear to break down organic materials. Okay, so basically what the lysosome does is it breaks down the organic materials which penetrates inside the cell. How? Through enzymes which they contain. Now, they have different names. Like, this is important. Like, you can get a question like that: who is called the digestive bag, or the demolition squad, or the suicide bag of a cell? 1. Lysosome, B. Ribosome, C. Mitochondria, D. Cytoplasm. This kind of questions can be asked in both UPSC and WBCS, because UPSC is changing its pattern nowadays. We can see that. So the first thing is, why this is called digestive bags? As they destroy foreign materials. You have to know the reason behind why they are named like this. So they are called the digestive bags because they kill the foreign materials. They are called the demolition squads because they remove the cell debris also. And they are called the suicide bags because they may burst when the cell gets damaged. Like when the cell dies, okay, or the cell is no longer capable of doing the work, that time the lysosome burst out, and that is why they are called the suicide bags. So these are the three very interesting facts about lysosomes which can be asked in the question. Moving on to the next organelle which is important, that is mitochondria. We have always known about the mitochondria, that this is called the powerhouse of the cell, or in Bengali we say it kosher shakti ghor. So what is this? The double membranous structure of which the outer membrane is porous and the inner one is found in folds called cristae. This is the double membranous structure. We have seen this is a kind of a, if you just if you have ever had sausages, then this looks like a sausage with some kind of stripe inside it. So this is double membranous structure, okay, and the outer membrane is porous and the inner one, which it can be folded. The next point which is important, what is this? The site of cellular respiration, where carbohydrates and fats are oxidized to release energy, which is used to form ATP. So actually when the carbohydrates and fat goes inside the cell, this mitochondria collects it and it oxidizes it, and after that we get the energy from the cell and we get the energy for our body to work. That is why it is called the powerhouse of the cell. But this thing is absent in bacteria and mammalian RBC as well. Okay. Now let us move on to the plant cell specific organelle, that is plastid. What is plastid? Plastid is nothing. Plastid is actually, you can say, determines the color of flower and the leaves and everything. Found in most plant cells but are absent in animal cells. That is why we don't have the plastids. Okay. Like the mitochondria, they have also their own DNA and ribosomes and are self-replicating as well. So they have also their own DNA. Like mitochondria has its own DNA. That is why it can replicate itself. And mitochondria has also its own ribosome. So basically plastids are kind of a mitochondria of plant cells. There are three types of plastids. First is chromoplast. What is that? Colored plastids except green. Imparts colors to flowers. What I was telling you, that plastids, they define the colors of a plant, of the flowers, of the leaves and everything. So chromoplast, they define the color of the flowers. Leucoplast, this is a colorless plastid, store food in the form of carbohydrates, fats and proteins. So leucoplast is essentially is responsible for storing the food value inside the cell, which we get from the plant. And lastly, the chloroplast, which is the most interesting plastid among the three, that is the green plastid. This plastid actually contains the chlorophyll, and that is why we see the plants are doing the photosynthesis. This is found in green algae and higher plants, definitely. This chloroplast has two distinct regions. One is grana and another is stroma. What is grana? Stack of membrane bound flattened discs containing chlorophyll. So this is actually, you can say, a sac or a stack of membrane, okay, which contains the chlorophyll. And stroma, homogeneous matrix in which grana is embedded. Now inside stroma the grana stays, and inside grana the chlorophyll stays. So this is the structure of plastid, and this is how the plant does its work of photosynthesis. It gets its color and it stores the food value through the plastids. The next organelle is vacuoles. What are vacuoles? These are solid filled or liquid filled small rounded bodies. Okay, either it is filled with solid things or liquid things, bound by a single membrane, which helps maintain the osmotic pressure in a cell and gives rigidity and turgidity into the cell. So actually, for doing the operation of osmosis, or taking the fluid or the foods or water from the outside of the cell, we need these vacuoles. In plant cells, these are large and permanent and almost occupy the entire region of the cell. In animal cells, vacuoles present are oxysomes. Now, what is this peroxisomes or oxysomes, you can say? Mostly found in kidney and liver cells. Okay, what are these? Small organelles containing powerful oxidative enzymes. The name itself suggests the work of this cell, oxysomes. It means it has oxidizing or oxidative enzymes, specialized for oxidative reactions such as detoxification. Means like if you had, like suppose you have had some kind of a very toxic element, okay, very rich food for one night, so the body has to get adapted to it. Body has to just separate the waste materials or the what should I say, the harmful materials from your food items, and body will extract only the solid and good item for your health. So this oxidizing enzymes, which is present in oxysomes or peroxisomes, which is present in kidney or liver cells, they detoxify the food items, and after detoxifying, the waste material that goes, the debris that goes out, and by lysosome definitely, and the other things stay. So this is the work of oxysomes or the peroxisomes. Now we have come to the main item of our, or main organelles of our cells, which is nucleus. Without nucleus, the cell cannot exist. Like without sun, the solar system cannot exist, or maybe the solar system exists but the life won't be. Anyway, so what is nucleus? This is the centrally located cellular component. Okay, this locates in the center, which I told you it's a kind of epicenter, bound by two nuclear membranes that enclose the nucleoplasm and separate it from the cytoplasm. There's a two nuclear membranes. These membranes, what does it do? They actually separate nucleoplasm from the cytoplasm, controls all the metabolic activities, whatever we are eating, whatever the metabolism rate we need, it controls everything, regulates the cell cycle, is kind of a master of the cell, you can say. Two structures are found in nucleus. What are those? One is nucleolus, that is site of ribosome formation and is rich in protein and RNA and chromosomes, okay, or chromatin materials or chromosomes. So one is nucleolus and another is chromatin material or chromosomes. Present in nucleus and visible only during cell division. These chromosomes, these are present in nucleus definitely, but this is only visible during the cell division. We will be reading when we will be doing the cell division thing, this meiosis and everything. We'll be looking at this how the chromosome actually comes during the cell division and everything. Contains hereditary information in the form of genes. These chromosomes definitely they contains the hereditary forms, like how we look like our grandfathers or our fathers. Like we often say that you have your mother's eyes. Like if you are a great reader of Harry Potter, you might get this reference every time that you look like your father and your eyes are of your mother's. This is Harry's natural description whenever any contemporary of his father sees him. So where does he get all those things? Those traits of genetic resemblance, they come from chromatin materials only. They come from chromosome only, and this is made of DNA and proteins. So nucleolus, this is made of RNA and protein, and chromatin material, that is made of DNA and proteins. Okay, now we'll go to the chromatids. So the chromatin or chromatids, what is that? A cell duplicates its chromosome before a cell divides. Definitely, otherwise how will it divide and how will it create another cell? So it duplicates its chromosome before a cell divides. When the two copies of the cells, which one cell creates another copy, are connected to each other or are attached to each other, they are called the sister chromatids. Okay, both of them are definitely identical and have identical genes. Definitely they have. Now when the cell division happens, what does it do? The sister chromatids separate from each other and two independent daughter chromatids comes into form. Okay, so the sister chromatids, they separate and they become two daughter chromatids. Now what does it happen? Now what happens? Now number of chromosomes may vary from species to species. Okay, definitely, like the chromosome in me will be varying with the chromosome of a cat or chromosome of my pet cat. Now human beings, they have 46 chromosomes. We have 46 chromosomes, 44 autosomes and 2 sex chromosomes. These 2 sex chromosomes, they define our sexuality or the gender in. Okay, now there is always a pair of chromosomes of each kind, like XX, XY, YY. This kind of pair is there. Okay, there will be always a pair. The paired condition of chromosomes is known as the diploid, and the cells that have the full number of chromosomes is called the diploid cell. That is the body cells in human beings. So the paired condition of the chromosome, like XX or XY or whatever the pair is, that is called the diploid, and the cells, those have the diploids, are called diploid cells. This is a very easy concept. And cells that have unpaired chromosomes of each kind are called haploid cells. That is gametes or sex cells in human beings. Okay, those cells which don't have any kind of paired chromosome, those are haploid. So body cells in human beings, those are diploid. Gametes or sex cells, those are haploids. So these are the main organelles of a cell which we have discussed so far. Now we will be going to the cell division and its theories. Like there are 2 theories, that is mitosis and meiosis, and after that we will be very shortly looking up to the matter of the difference between 2 systems or the 2 processes, not system, I'm sorry. And after that we'll be going for one question and our chapter will be over. So I hope that till now you have liked our presentation, and if you have any kind of doubt, please do ask, and we ensure you that once we have started this series, we will be continuing with this series in English. And if you want any of the part of this series in Bengali or in bilingual, then do let us know. We will be trying to do as best as possible. Now moving on to the process of cell division, we have two processes which is very easy to understand and very easy to remember as well. One is mitosis and another is meiosis. Now what is mitosis? Mitosis is a form of eukaryotic cell division, okay, that produces two daughter cells with the same genetic component as the parent cell, okay. So this is a kind of a process of eukaryotic cells which actually produces daughter cells from the parent cells. Now how does it happen? We can, like we are showing you here the process of happening over here. Like the chromosomes thickens, okay, the chromosome comes together and thickens and shortens, and the nuclear membrane breaks down and meiotic spindle is established. This is the first phase, that is called prophase. The second phase is metaphase. What happens in this phase? Chromosomes are aligned in the middle of the spindle and they split longitudinally. Now look at the process of this metaphase. You can see that there are some kind of longitude which has been drawn over here, and the chromosomes are aligned along with that in a very, the middle of the spindle, of the longitudinal spindle, and they are ready to break. In anaphase, what happens? The chromatids travel towards the opposite poles. Now the chromosomes, they break, the chromatids, they travel towards the opposite poles. And in telophase, what happens? The chromatids begin decondescending. Like the chromatids, when the chromatids are reached on the two different poles, then they begin to decondescend, they begin to break, and they separate from each other. This is the process of mitosis. This is how the eukaryotic cell does separate from their parent cells. And the next process is meiosis. What is meiosis? Meiosis is the form of eukaryotic cell division that produces haploid sex cells or gametes, which contains a single copy of each chromosome, from diploid cells which contain two copies of each chromosome. Basically, it transforms the diploid cells to haploid cells, okay. And this process takes the form of one DNA replication followed by two successive nuclear and cellular divisions, okay. That is meiosis 1 or meiosis 1 and meiosis 2. We don't have to go into the details of this thing. We just see the phases of here. The first is interphase, okay. We can see the centrosomes over there. Then the prophase comes, and spindle begin to just take into place. Then the metaphase comes, the spindle spreads more. Then the anaphase come, the sister chromatoids, they remain attached to each other, and then homologous chromosomes separate, okay. So this is the process through which the meiosis process, through which the cell gets divided. And this is more complex, definitely, because in mitosis we saw that the process was much simpler to understand, and in meiosis we can see that this happens in a very complex and very difficult way. But thank God that we don't need to understand all those difficult processes over here, because we don't need all those from the point of view of exam. Next, we will very shortly just understand the difference between two processes. One is mitosis and another is meiosis. First, mitosis, it takes place in somatic cells. Meiosis occurs either in sex cells or during zygote germination. Mitosis, single division procedure, 2 cells. Meiosis, double division produces of 4 cells. So this is single division procedure, it produces only 2 cells. That is double division procedure, it produces 4 cells. Mitosis is comparatively simple. Of course, meiosis is complicated. In mitosis, a cell can undergo repeated mitosis, but meiosis occurs only one. In mitosis, number of chromosomes remains the same after the mitosis. Because it replicates. In meiosis, the number of chromosomes are reduced half, because with the starting of the process of meiosis, the chromosomes get In mitosis, it takes part in healing and repair. Meiosis, it takes part in gamete formation and maintaining chromosome numbers. In mitosis, no variation is introduced, or no variation are introduced. In meiosis, it introduces variations. So these are the very basic differences between the two processes. I hope that you liked this video. I hope that you understood something from this video. And as we have told you that there is one question which came this year UPSC, that is, which of the following statements are correct regarding the general difference between plant and animal cell. That is why I told you during the first during the first few minutes of this lecture that listen to this part carefully, because the question came. The first is, plant cells have cellulose cell wall whilst animal cells do not. 2. Plant cells do not have plasma membrane unlike animal cells which do. 3. Mature plant cells has one large central vacuole whilst animal cells have many small vacuoles. In this case, 1 is definitely true, because cellulose cell wall, any kind of cell wall, is not present in animal cell. And plant cells do not have plasma membrane. This is not correct, because plant cells, they also have plasma membrane. And the third one is, mature plant cells has one large central vacuole. Of course, definitely there is one large central vacuole. On the contrary, the animal cells, they have many small vacuoles. So here the option C, that is 1 and 3 only, that is correct option. So I hope that you like this video again, and we do love to continue this series unless we finish the whole syllabus. So please stay tuned with us, support us. We will be back with the next part of the biology series very soon. For that, we need your support. Do like, share and subscribe our channel. Like and share this video, and press the bell icon for the more updates. Thank you very much. Stay well, stay safe.